Right. Can you see my slides now?
Yes, we see it.
Okay, great. All right. So coming back to modeling, this is work we've been doing for a few years now, and it's a collaboration with Kevin Urdel's laboratory at Seattle Children's Research Institute. Short outline of my talk. So I'll give a little bit of introduction about tuberculosis, and then I'll kind of tell you short two stories where we try to understand dissemination of two strains of MTB, Erdmann and H37RV, and then finish with some conclusions. So tuberculosis, I hope people have heard that this is disease caused by Mycobacterium tuberculosis. These are bacteria. One interesting observation about MTB or Mycobacterium tuberculosis infecting humans is that if older studies that looked at deceased humans that died of reasons others than tuberculosis, and if you look at scans, so X-ray scans of their lungs, many people actually had lesions, healed lesions, most likely caused by Mycobacterium tuberculosis. And if you look at locations where these lesions were located, right in the lung, they seem to be randomly distributed. So this is a composition of multiple scans from many people, and the dots represent where those healed lesions were found. So these people died for some reasons, but they were exposed most likely to MTB, did not progress to the disease, and then died, right? And so the lesions that they had, so this asymptomatic, I guess, individuals were randomly distributed. But if actually you do another study and you look at individuals who have active tuberculosis, and you ask the question, okay, where are the lesions where this Mycobacterium tuberculosis located? And then these people have a disease. You actually will find that they're not random. They're typically located in the upper parts and back parts of the of their lungs. So this is kind of, the head is facing that way. So it is upper and back part. So it's actually a big question. So if the initial or some deposition of the Mycobacterium tuberculosis when somebody inhales that is kind of random, why would somebody has a disease when it's specifically located in one particular part? So is that because some individual just unlucky and the bacteria when they're inhaled just come into that particular area and they cause the disease? Or is that the bacteria land somewhere in the lung but then somehow disseminate to that place, to that upper parts of the lung, and then as they disseminate, that causes the disease. So this is one of the big questions actually in tuberculosis. Like, we still don't understand how exactly people develop the disease, right? And another complication is that if an individual is immunocompromised, for example, HIV infected, or maybe has cancer, MTB may be found outside of the lung. So even though it's pulmonary disease, typically tuberculosis, MTB can spread to other tissues in the body, and it's called then extrapulmonary TB, and may affect bone marrow, can spread to the brain. How exactly, again, the bacteria disseminate from the site in the lung to those distant sites in the body is also largely unknown, how exactly it happens. So we typically don't really work with humans just because it's difficult to trace early events of MTB infection in humans. So we use mice. If you infect mice with Mycobacterium tuberculosis, you will almost always see dissemination of the bacteria outside of the lung. And it's you can do that if you infect mice with a so-called conventional dose. So if you aerosolically infect mice with about 50 to 100 bacteria or colony-forming units, you will see dissemination. Or if you use a lower doses, or a dose we call ultra-low dose, which is a few bacteria, 1, 2, or 3 bacteria, that is has been developed by Kevin Urdel's lab in Seattle. So again, independently of how you infect the mice, if you wait long enough, so typically takes a few weeks, you will find the bacteria outside of the lung. you'll find in the lymph nodes, in the spleen, and also we recently found them also in the blood. So we would like to understand how this dissemination occurs. We are not first to ask that question. In fact, this particular study has already kind of asked that question. Well, how do bacteria, after initial infection of the lung, disseminate to other tissues? So they did the following experiment. They infected mice with a relatively low dose of a particular MTB strain called Erdmann. They didn't really quantify the dose, so we don't really know how many bacteria were inhaled by the mice. And then they measured at different time points how many bacteria are found in the lung. Then they looked at pulmonary lymph nodes, so lymph nodes associated with the lung, and in the peripheral tissue, such as spleen and liver. And they counted how many bacteria are found in these different locations at different times. And they proposed that their data is consistent with the kind of this particular pathway. So initially, bacteria are in the lung, then they disseminate into pulmonary lymph nodes, and then from lymph nodes, they seed into the peripheral tissues, but as well as perhaps lung also has some distribution of bacteria into the peripheral lymph nodes. And then you have also migration of antigen immune response or to the bacteria. So the question I'd like to pose is that, is the proposed dissemination pathway correct? So this picture that was drawn, is it correct? So, and I think this is, again, an example of how you can use mathematical model or modeling, is that people make cartoons like these based on data all the time, but whether these cartoons are actually correct, we don't know. So you can use mathematical modeling to ask the question, is this particular cartoon consistent with the data that somebody actually has? So here's the data that was generated, and the authors of that study shared those data with me. So they did an infection in 2 strains of mice, B6 mice. These are more resistant mice to MTB infection typically, and C3H mice. These are more susceptible. They develop much more severe disease. And so they measured bacterial numbers in the lung, lymph nodes, spleen, and liver. And at these particular time points that you can see, and you can see accumulation of bacteria in the lung, in the lymph nodes, in the spleen and liver, and the same thing for C3H mice. So we wanted to ask a question, okay, so if this is the data, let's see if we can develop a model that would describe dissemination kinetics of bacteria between the tissues, but we will try to formulate kind of alternative models that would either model direct dissemination of the bacteria. So in this particular case, bacteria replicate in the lung and then disseminate to the lymph nodes or spleen and liver. So directly, there could be indirect dissemination. The first tissue that the bacteria disseminate from the lung is the lymph nodes, and then they go systemically to the spleen and lymph node, spleen and the liver, or maybe alternative indirect direct dissemination, where first bacteria actually spread systemically, and then they infect the lymph nodes. Right. So these are 3 models that we proposed. And then these cartoons we can describe as a system of differential equations with different rates of migration between the tissues. And this is the full model that assumes all the different dissemination events. But then if we say, if it's only this particular, for example, model, direct dissemination, that we say from the lung, you only go into, say, lymph nodes, spleen, and liver, but you don't, from the lymph nodes, you don't go anymore into another tissue, for example. And so I like asking questions when I do presentations. So given this data and given this schematics and given this cartoon of a mouse where the tissues are located, which of the pathways do people think is the most likely to explain this data? So I'll give you a little bit of time to think about it. I mean, we can do a chat vote. So dissemination pathway is direct dissemination, indirect dissemination, or alternative indirect dissemination. And these are how they would occur. No answers? Well, typically somebody answers. Okay, we're moving on then. Nobody answers. But well, the answer is simple. Actually, all of these pathways are consistent with the data. In fact, we couldn't discriminate. So, for example, this is one of the pathways, and you can see that the model fits the data very, very nicely for CFU in the lung, CFU in the lymph nodes, CFU in the spleen, or in the liver. But if I take another pathway, it also fits very well. So this pathway is when we disseminate from the lung to the spleen, and from the lung to the liver, and then from the spleen to the lymph nodes, and or from the liver to the lymph nodes. And then there's another one. So here's another pathway. Here's the parameter estimates for these three alternative models, and we can evaluate the quality of the model fit based on sum of squared residuals, which you can see they're very similar, a little different, but really similar. And then we can also calculate so-called archaic information criterion, which takes into account complexity of these models. And these numbers, again, are again very similar. They're not dramatically different. So the point here is that the modeling doesn't really tell us what the pathway of dissemination of the bacteria is. But the modeling can tell a little bit we can make some observations. For example, what is consistent independently of the model, so we can take different pathways, but what we find consistently is that the replication rate of the bacteria in the lung or in the other tissues is consistently higher in black 6 mice compared to C3H mice. And that's kind of a little interesting because C3H mice are susceptible mice. So these are the mice that typically will die faster and with stronger pathology compared to B6 mice. But actually, the bacteria replicate at a slower rate, which is kind of unexpected. And then this is for another model. So this is one pathway of dissemination. This is another pathway. We find the same consistent lower replication rate. However, the rates of migration between the tissues, even though they were dependent on the model, they were not very much different between the strains. So something is different between these two mouse strains is that the bacteria replicate at different rates. But the dissemination occurs at least similar kinetics. So first part, concluding, something that in the modeling people typically we want to find what our best model, what is the best mechanism, what explains the data. And unfortunately, in this particular case, we couldn't tell. We couldn't actually say that this pathway or this pathway is more likely because the alternative models fitted the data with similar quality. As I just said, the replication rates are lower in C3H mice, which are susceptible mice, compared to black 6 mice, which are considered to be more resistant. And they're not dramatically different, but there is 20% or so difference. An interesting observation we've made that when we estimated the rates of MTB replication is that MTB appears to replicate faster in the lymph nodes compared to any other organs like spleen or even the lung by a relatively big percentage, and it was consistent. Again, this result was independent of the model that we've tried. And it's surprising because, again, MTB primarily infects the lung, and you would think that lung is the tissue where the bacteria would replicate the fastest, but actually, at least in this data, it was not the case. We could actually suggest that one of these three models, which was called alternative dissemination model, is not actually probably physiological, because even though it fitted the data perfectly fine, it required very high rates of dissemination and high rates of bacterial replication in the liver and the spleen to explain the data. So, for example, rate of replication of bacteria in the spleen was estimated to be 5 per day, and we think that bacteria probably divide at least at most once a day, maybe even less than that. These are very slowly replicating bacteria. So another kind of take-home message here is that your model may be able to fit the data perfectly fine, but the parameters at which it's doing that are not physiological, and this would suggest that this particular model perhaps is not a good one. Again, even if we cannot pinpoint fully what the pathway of dissemination is, we can actually quantify how many bacteria go to different tissues, and we can do it per model. And what we find is that even in the model where the bacteria disseminate directly from the lung to other tissues, about 75% of the bacteria actually go to lymph nodes. So again, suggesting that probably dissemination to the lymph nodes is one of the primary pathways of the bacterial dissemination systemically. So, and that's something will come up at the end as I summarize. So another question I have, so here's my data that I showed. This is the data from the study, and the model fits, which I think are pretty good. But does anybody see any problems with the data, or particularly with the model predictions? Again, I'm asking a question. I don't know if anybody listening or could give me an answer. What do you think wrong with the model predictions here? Or nothing is wrong? It's perfect. Okay, I see in the chat somebody saying outliers maybe. Okay, anybody else? Chat works too, if you want to do it in the chat. Time. So time is the problem. It's always time. Probably is a problem. We never have enough time. I'm not sure I understand what time question mark means. It's imprecise. I'll give 5 more seconds. Time of infection. Okay, maybe that's kind of what I'm thinking. So if you look at predicted curves at the very first few days, you will see that there is some kind of weird delay, and then the bacteria grow. So the reason is because the model would predict that you should start with fewer than 1 bacteria. So you can see at the scale, this is 10 to the 0, so that's 1 bacterium. So in order for me to fit this rapid increase, I had to kind of assume that I start with very, very low levels of bacteria. And this is kind of weird because probably they started with, I don't know, 10 or 100 bacteria. So we don't know what the dose was because it wasn't measured in this experiment. So the problem is we don't have this early dynamics. So in collaboration with Cameron Urdal lab, a postdoc, and now she's a staff scientist, Lauren Cross, did these experiments. Essentially, she repeated the experiments that this previous study did, but she used a different strain, H37RV, and she used the conventional dose, although she got about, her deposition dose was about 10 bacteria per mouse. And then she measured how many bacteria there are in different tissues in the lung draining lymph nodes and spleen. So she didn't measure in the liver, but just in these tissues. But she did much more earlier measurements and for longer measurements. So we have many more data. And there were some other measurements done, including measurements of bacteria in the blood. I'm not going to kind of mention about that too much. And here's the data. So this is from Lauren's experiment. So this is how many bacteria you find in the lungs over time. And you can see how beautiful they look. You have seen increase, then you actually see a control of bacteria. And then by 100 days, there's some kind of recovery. This is the lymph nodes. And again, you can see accumulation, control, and maybe maintenance of bacteria in the lymph nodes. This is in the spleen. And this is detection in the blood using a specific medical system where you measure when you deposit the culture in the culture sample where you think the mycobacteria are, and then you measure time to positivity of when this culture, when this media, when you put the sample, becomes positive. And if it becomes positive before 40 plus days, it means that there were bacteria deposited. So we can actually measure how many bacteria, or not how many, but time to positivity from the sample of the blood. I'm not going to use that data here though. So we did the same thing. We essentially tested if we have direct dissemination model, indirect dissemination model, or alternative direct dissemination model. We don't have liver here, but it's going to be just spleen. Very similar models. And unfortunately, the answer was that all these models can fit the data perfectly fine. So again, we cannot necessarily, we can, even from this data, which are much better, and you can see the model fits kind of impressive, in my opinion. So we can describe the data very, very nicely, but we cannot tell which is the right model because they essentially, if you look at SSR, so sum of squared residuals, so the how much model deviates from the data, they're about the same, so almost no differences. One of the things that is a little problematic in this approach that I just did is that when we fitted models to data, we assumed that the model dissemination occurs kind of in deterministic manner because we used a system of differential equations. To investigate if the parameters that we estimated are kind of reasonable, I took this mathematical model and the parameters we estimated, but then ran the model stochastically using Gillespie algorithm and library Gillespie SSA2 in R. And then I asked well, will the model, if I run it stochastically, assuming that I start from 10 bacteria, because that was experimentally done in the lungs, will it describe bacterial dynamics? And I tried it for two models, both of this indirect dissemination model or direct dissemination model. And here you see all these gray lines are the individual runs when I run stochastically, and it generates a little bit of a spread, which kind of covers the data points, which is nice. And what I found really exciting is that I actually can now explain this variability in the data when the bacteria appear in the lungs, in the lymph nodes or in the spleen. And they kind of, the stochastic simulations kind of cover this data, suggesting that the variability that we see in CFUs can be explained just due to randomness of this dissemination event from the lung to the lymph nodes and to the spleen. So, okay, I'm finishing. So some of my conclusions. So first, one of the things I think is important when you do modeling is always consider alternative models and don't stick with one model. That's typical, that people just write one model and then work with it. But the problem of having alternative models is that it's hard to reject them. If your data is not great, or even if you have good data, sometimes the data is just insufficient, I guess. So we don't know still what is the exact pathway of dissemination is of the bacteria from the lung to systemic organs, but we think most bacteria most likely go through the lymph nodes before they enter systemic circulation. And that's the estimate is for H37RB. So my second part of the talk was about 85% or so bacteria go through the lymph nodes before they enter circulation, and perhaps about 75% for Erdmann, which kind of I think is exciting because again, it suggests how we could block perhaps migration. We have some data where we looked at flow cytometry. With flow cytometry, we looked at cells that are infected in the blood, and we think that majority of cells are monocytes and neutrophils. And we think that neutrophils most likely came actually directly from the lung because they have a phenotype of so-called reverse transmigrating neutrophils. And monocytes, we think they're probably coming from the lymph nodes. And so the pathways of how cells enter the circulation indeed could be different because we do find two different cell types that carry MTB in the blood. And again, finding that most bacteria enter the lymph nodes before they probably disseminate systemically suggests that if we control bacteria in the lymph nodes, then perhaps that will reduce ability of the bacteria to disseminate. There's a few limitations. I like always pointing limitations of what I do. And one of the limitations is that you notice in my model, I use logistic growth to describe the kinetics of the bacterial replication. And then even though it fitted the data perfectly fine, you can see that the model actually did not explain very high variability of bacteria that we find at about 28 days post-infection. So this variability I could explain in stochastic simulations, even this, but I couldn't explain this part. And I think that's because logistic model that I used is just not adequate for that purpose. Another thing that actually happens with bacteria, especially with Mycobacterium tuberculosis, is that they actually, after the deposition in the lung, they don't start growing right away. There is a delay phase. We got this interesting data from John Pearl from the UK, where he actually measured very, very carefully by infecting mice with about 44 bacteria or so, many mice, and measuring how many bacteria you find in the lungs over time. So this is linear scale for 150 days on the log scale. And what you can see here, even if when you look at day 1 or day 2 or day 5, you only see for a few first days bacteria don't replicate. Only at day probably 5 or so you start seeing them grow, and then in the next several days they really grow exponentially, very, very rapidly. So we didn't include that resting phase in our modeling. That's something that we need to do. And then again, this data that I showed before on MTB Erdmann infection, there was no early dynamics measured. So the first measurement they had in their data was on day 8. And then we may overestimate actually how quickly bacteria replicate just because we look at the very late stages of the infection. And there's a few future directions where we want to go. So we still want to understand how MTB disseminates specifically in the blood. So how to use this metric of time to positivity from the assay that I mentioned. It's called MIDGET. And so we have some results, but they're not final yet. We want to understand whether the bacteria disseminate differently in this conventional dose infection, which is about 50 to 100 bacteria, versus so-called ultra-low dose infection, where you infect typically with 1 bacterium per mouse. We need to go to more mechanistic models from these logistic models of MTB dynamics, just because we know there's more cells involved in MTB replication, like macrophages, neutrophils. And we know the immune response needs to be included as well. We know what controls the bacteria at the current kind of capacity. It's most likely immune response, and then including that would be very useful. Lots of acknowledgements. So Sam Beher shared the data from his publication that I showed on MTB Erdmann. Lauren Cross did the experiments in Kevin Urdel's lab with MTB H37RV. Lots of feedback from Erdal lab members about TB pathogenesis. I know more about it now, but I continue to learn, and NIH for funding. And that's it. I will stop here and I can go back if anybody has any questions. And I know for you guys it's been a while, and then it's been a long day for me too.
Thank you, Professor. I have a question. Do you know any
Sure.
factors affecting the decision of dissemination pathway?
Say it again. Do I know any what?
Any factors affecting the decision of dissemination pathway? I mean,
The biggest factor is dose. So the more bacteria you would have initially, the more rapidly they'll disseminate. So we have the data where we infect with ultra low dose, and dissemination is actually delayed substantially, and then in the blood you don't always detect bacteria. Anything else? Don't know. Actually, that's a good question. I mean, we know for humans, if you're immunocompromised, so again, chemotherapy, if you have cancer, for example, or if you're HIV infected, so you are more likely to have extrapulmonary tuberculosis, so having sites outside of the lung to be infected. So if you're immunocompromised, it's more likely you're going to have disseminated bacteria. But is there something else, like, I don't know, diabetes, or is there, I don't know, larger body weight or something? Don't know that part. Yeah.
So some viruses and bacteria have different variants. So can variants also be impacting that?
Can variants what?
Certain viruses have different variants. So can different variants of the same virus impact the dissemination pathway?
So, I mean, sure. I mean, I don't know. I don't have a good answer on that. I mean, in this case, it's bacteria, it's not viruses. I did compare two strains, right? So, Erdmann, MTB Erdmann. So these are two strains, and H37RV, that's another strain. So, um, again, I cannot say if they disseminate differently because I couldn't really establish the exact pathway of dissemination. I can say that there may, but the rates are different, definitely. But the pathway itself, I cannot conclude that they're different because I couldn't say for sure what's their pathway.
Thank you, Professor.
So, yeah. Mm-hmm.
Any other questions?
I have a quick question. You were saying that the logistic model was not good, and you did some stochastic simulations, and you showed that ash lines that covers the data in the increasing area. Yeah, these ones. So I'm curious whether if you can do a uncertainty quantification to figure out what's missing in the model, and then you can have a direction to do a better model.
Mm-hmm. Yeah. Well, I mean, I know why. I don't need to do the quantification, uncertainty quantification. I understand why the model doesn't generate statisticity. So what happens in the logistic model is that when you approach carrying capacity, and here you approach in all these states, you approach carrying capacity, right?
Mm-hmm.
So what happens is that you don't allow any stochasticity in dynamics because uh, because of the carrying capacity, because it fixes you to one value.
Right.
So, a trajectory that went lower will still reach the same carrying capacity. So the solution here is that there should be some kind of a time dependent. So carrying capacity has to be like time dependent. Then you would generate more variability here. So you would reach higher levels for bacteria that disseminate earlier versus the ones that disseminate late. You should have lower levels. And the way you can do time-dependent carrying capacity is actually immune response. So we are working on that. I mean, I have a version of a model that it's a time-dependent immune response that controls the bacteria. And so that immune response will control bacteria depending on their density. And it can generate this variability at the later time point. I have a working model. It's not, I need to put it together for the presentation mode, but I have something that is working. Yeah.
Okay.
But that's something. This is important for modeling again. If you find some particular features that your model doesn't fit the data properly, or has some elements that doesn't. So this is very, very important to think about. Well, how can you change the model? And that's something that we don't really teach very well. This is very difficult, actually. So you have to have a lot of kind of intuition. Is like, well, what drives that particular phenomenon? How could you change it? So that one is not always easy.